Merci. Je suis très heureuse d'être ici et avec votre permission et collaboration, je vais vous donner mon texte, mon présentation en anglais. I just think better in English, I'm sorry. Mais j'ai des diapositives en français. Ça c'est mélangeant. So what I want to talk to you today is about synthetic biology. I am an evolutionary biologist who doesn't really do this kind of work specifically, but I can sort of, I know the science well enough to be able to explain it, and I've been watching Regarder l'espace for a few years now. So let's just begin. What is synthetic biology? Fundamentally, it's applying engineering principles to biology. There's, I don't know if I have, well, I don't really need a pointer, but anyway, there's really two flavors of this. On your left is what we call the biobrick idea, which is basically stitching together pieces of DNA to make something. Each piece has a function. The red part, for example, says start here. The yellow one, make something interesting, maybe a molecular compound like vanilla or something like that. The green says I'm doing this. The blue says this is an antibiotic resistance or something like that, to say this is how we're going to keep it in the space that we in the cell that we need it, and then stop doing it. So you have a biological device. Just like with the internet, the design of the internet, or any engineering or any other kind of engineering feat, you have pieces that you put together and they function. The second part is genome editing, which I'll talk about a little bit more about in a minute. And you've probably heard of, especially very recently. A couple days ago, there was a discussion of a Chinese researcher who apparently had edited the genome of twins to be HIV resistant. The key idea behind synthetic biology is that the genetic code can be written, not just read. And that's the most important part. Two features have been driving this process. One is the reduction in the cost of DNA synthesis. And that's happened alongside, at the same time, as the reduction in cost of DNA sequencing. Okay, so in blue, you see the cost of sequencing from about 1990 down to 2017 dramatically drops. I mean, I'll tell you, this has revolutionized my own career. When I started at the University of Ottawa in 2003, I never thought I would be able to actually get access to the genome. I had a crazy idea with another postdoc about how to do that in a very clumsy way. And within 7 years, we could do it. Now it costs me about 100 Canadian dollars to sequence one microbial genome. It's incredible. Parallel to that, in red, you can see the decreasing cost of synthesis. It's still orders of magnitude higher than sequencing, but it's coming down all the time. So that's really what's driving this. The second is some new discoveries from nature and and elaborating some technologies around this for genome editing. You might have heard of CRISPR, which stands for clustered regularly interspersed short palindromic repeats. It doesn't really matter what it means. Or what it is. CRISPR-Cas9 is basically a protein DNA complex that goes into a target area in a genome, cuts it, and provides an opportunity for new genetic material to be inserted. It's a bit like renovating your apartment or your house. You have a structure already there. You'd like to improve, say, the bathroom or the toilet or the salon or whatever it is, and you just go in and you do some modifications and then off you go. You've still got more or less the same thing. But you can do that in any kind of target cell, whether it's human cells, zebrafish, or bacteria. Just a little animation to illustrate how CRISPR works. This is a text from Genesis 1:28. Be fruitful and multiply and fill the earth, which is not good English, and subdue it, and so on and so forth. Well, here comes the CRISPR complex, CRISPR-Cas9 complex, with a complementary sequence here, removes it, and we insert the correct word now. That's basically how CRISPR works. So these technologies, synthesis and gene editing, are being used in a variety of different ways. on the left, the most common is to produce interesting things, much in the same way that we would produce other kinds of fermented food products, wine or beer or yogurt or anything like that. But they're used specifically to produce a particular kind of product, vanilla, hormones for, say, sex and gender transformation, or even possibly toxins. But let's not go there just yet. CRISPR technologies are being used in a variety of ways. I mentioned the Chinese researcher from a couple of days ago, but the most common one that people are thinking about right now are what are called gene drives. This is manipulating the normal sex ratio of, say, mosquitoes from 50-50 male-female and biasing that towards one gender or the other, which results in the crash of the mosquito population, and thereby potentially controlling malaria in areas where it's effective. You get rid of the mosquitoes, you get rid of malaria. I think very interestingly are a couple of other, on the right here, a couple of other technologies that are basically surveillance techniques. The bottom one is from a colleague of mine, Alexandre Poulain, who has designed a microbial bioreporter to detect arsenic in water, in soil water. So basically, it's a short bit of DNA, a device that's inserted into dried bacteria, into bacteria. They're freeze-dried. He takes them with him into the field in Cambodia or Vietnam or wherever, and he has a 3D printed spectrophotometer with him that some engineering students at the University of Ottawa designed. And you add the water. If it glows green like this, you've got arsenic, and the amount of green indicates how much arsenic you've got in the water. The other one on top is a paper-based detection of Ebola, which is effectively, so what you do is you print DNA sequence unique to the Ebola strain onto a piece of sterile paper. You take that with you into the field, take a sample, and if it turns purple, I think, yes, if it turns purple, it's Ebola. If it's not, so it's a bit like litmus paper. This is quite incredible because it really reduces the cost and time associated with detecting the kind of pathogen you have. There are challenges with all of this. There are knock-on effects that are not well understood with CRISPR. But importantly, I think there's one which sometimes is called the technological debt between evolution, what evolution has produced, and what we can engineer. Evolution has produced things over billions and billions of years that work really well and are quite robust. The devices, the biological devices that we engineer are designed by us, and they're not necessarily that robust. They're quite fragile, in fact. There's a second issue, very important in the innovation space and entrepreneurial space, which is how do you scale up these processes to be economically viable? But what's happened, importantly, is that despite all of those challenges, the cost of entry, the barriers to entry are falling in this space. And so we've seen the rise of what we call do-it-yourself synthetic biology. So on the right, or on the left rather, is an example from here in Paris, La Paillesse, which is a community-based synthetic biology lab. And on the right is a picture of one of the latest IGEM, or International Genetically Engineered Machine competitions from their big global jamboree. It basically means that anybody can do this anywhere at any time for relatively little money. You can order CRISPR kits online for about I think it's $100 American or something, and delivered right to your home. So it's important to ask, who's actually doing this work? This is a slide summarizing the results of a study published in 2013, so a bit out of date now, by the Synthetic Biology Project at the Woodrow Wilson Center at Harvard, who surveyed American people, the American community of DIY synthetic biology. Typically, the people doing it are under 45, so they're fairly young. Okay? They don't necessarily have this sort of license to practice that comes with advanced training at the PhD level in research. They're very often, say, bachelor's level undergraduate students, sometimes just high school graduates. But they often work in collaboration with experts, because doing this stuff it sounds easy, but it's not. So they need that guidance. Because they're young, they tend to be very innovative, entrepreneurial, and they are really keen on peer-to-peer education. So there's a democratization of knowledge, the spirit of democratization of knowledge here going on. They're working principally in established laboratories, also in community spaces like La Payas, universities, industry, government, and so on and so forth. There are some who are working out of their kitchens and garages. So that's what we know. This just illustrates the increase in the DIY bio space, especially in the US and Europe. On the left, you can see since about 2005 up until 2017, a massive increase in the number of people. I mean, I shouldn't say it's a massive number of groups, it's just a fast rate of increase. And on the right, you see the growth of the iGEM competition. The total numbers of teams is now in the 100, I think it's about 400 at the last Jamboree. In green, you can see the increasing numbers of high school teams as well. So let me summarize this by saying that the promise of synthetic biology is not just what can be produced, but it's who is producing it and where they're producing it. And those are the perils or potential perils or the challenges. There are two major implications. One is, you might call the social implication, is that innovation can in principle come from anywhere, right? From somebody's kitchen, and increasingly we're seeing the generation of startup companies, basically synthetic biology for hire. So you have an idea, you'd like to have a blue rose, a genuinely blue rose, and you say, I've got some money, I'm going to hire you, please do this for me. And they work it out, whether with whatever techniques and tools they have at their disposal. Here's one example, and you can see some others at the bottom. The second, and the one that gets many people anxious, is that the risks are highly distributed. So there is the potential for committed bad actors to do something nasty. Now, the community of DIY synthetic biology is very concerned about this, and they've developed their own codes of conduct. There's one for North America, there's a slightly different one in Europe. They don't generally want to be, they just want to do their stuff. They don't want to be bothered by anybody, and so they're willing to do the right thing. And they don't necessarily know what they're doing, and so they're willing to work with experts and with regulators and health and safety officers and so on. But if somebody really wanted to, they could order a bunch of parts from different sources, have them delivered, have them stitched together, in fact, and cultivate something very nasty in their kitchen or their garage. There's also ecological consequences that we really don't still understand from the accidental or intentional release of any of these products. If we get rid of the mosquito populations in a malarial infected area, that's potentially good for malaria, and it's probably good for the health of the human population. And so there's maybe it's the right thing to do. What we don't know is what the broader ecological consequences are going to be. How are the bats going to do, for example? What does that mean for ecosystem functioning? These are open questions, and it may be worth taking the gamble, but it is something to consider. And so I think it's important, I'll just end with this last slide, where I've got a few suggestions on ways in which we can start thinking about managing this space. The risks associated with DIY synthetic bio are not too dissimilar from the computer hacking sort of world. A little bit, there are differences. It's much harder to do this. You need also a base of knowledge to be able to do it. But here's some thinking, just 3 thoughts that I've come up with over the last few years of thinking about this. One is engage directly with community labs and the iGEM competitions and others. And in fact, the FBI in the United States has already done this. They've assigned one of their agents to be responsible for synthetic biology, and he basically goes around and just makes friends. You know, he's seen as he's not there to do anything else other than just figure out who's there, and to make sure that they know that there's a relationship. He's building relationships. You might take a lesson from the free economy model and give the community what they want and need. Give them infrastructure, give them tools, give them expertise in exchange for some data on who's actually doing this, right? Who's involved. And we might want to think about developing a capacity to trace back bits of DNA so that we know where it comes from and where it's going, who produced it and where it's being delivered. It's sort of an IP address for bits of DNA, a DNA barcode. That would help in the event of an accident or an intentional release of something to be able to figure out where this stuff has gone and who's doing it. There are ways of getting around that. The DNA, the genetic code is redundant, of course. But it's worth giving some thought. And in fact, some of the iGEM teams have started to do this themselves as part of their projects of thinking about safety and associated strategies with this. So I'll end there. Thank you very much for your attention. Merci beaucoup.
We have perhaps 1 minute or two for short questions. It's in the 20 minutes, so if you have any questions. Yes. So this gentleman here.
Concerning the last sentence, to retrace things for CRISPR-Cas9 is impossible to make this kind of things, and you can use CRISPR-Cas9 to avoid to have this kind of procedure. So it's a real problem, I think.
It is. What's helping us right now is that CRISPR-Cas9 doesn't work that well yet. But that doesn't mean it won't change within the next few years. I mean, it can work. It's really an issue to do it at scale. So it's great for researchers, but not necessarily yet at scale.
Next question here.
I'm not sure what you mean by the model of free economics. That is, in free economics, does it companies take into account their social and environmental responsibilities, or does it mean that it is out of their freeness?
I mean like how we all give away our data in order to connect to the Wi-Fi at Charles de Gaulle, for example. So we don't have to pay anything for that connection, but what we give up is who we are or where we're coming from. So that's what I mean.
All right, thank you very much, and let's go to the next speaker.